Assalamu alaikum dear students. This video lecture has been made for botany students who are in the 5th semester and have opted seed technology as a single course. In the syllabus of seed technology, the first topic in the first unit is seed development, and in bracket there has been mentioned as sporogenesis. So first of all, we have to discuss how the development of seed takes place. Now we will discuss what is actually seed. Seed is an embryonic plant. So we can also say that seed is a future plant. And where from this plant comes from the seed. So first of all, we will discuss the constituents of the seed. Seed constituents. Seed has three important parts. First of all, we will discuss three parts that are present in a seed of dicot or monocot plants. The first part is embryo. Its second part is endosperm. And its third part is seed coat. So three parts that are present in dicot or monocot seeds are embryo, endosperm, and the seed coat. Where from this tiny plant, when we sow any seed, that is dicot seed or monocot seed, where from the plant comes from? It is the embryo that gives us the tiny plant. The tiny plant is given by the embryo after we sow the seed in soil or any other suitable medium. Now, how the development of this monocot or dicot seed takes place, we have to follow the sporogenesis. Means we must know what is sporogenesis. So we will discuss here sporogenesis. So sporogenesis is a process in which spores are formed. So it is a process of spore formation. So process of spore formation is called sporogenesis. But there are two types of sporogenesis. Two types of sporogenesis are there. One is called as microsporogenesis. Microsporogenesis, and second one is megasporogenesis. These two processes are important. These are actually the two key steps that are responsible for the formation of, for the development of the seed. First is microsporogenesis and the second is megasporogenesis. First of all, we will discuss the microsporogenesis process. Microsporogenesis. In this term, microsporogenesis, there are two components. One is micro, another is sporogenesis. Micro means, micro here means microspore. Or this microspore is also called as pollen grain. This microspore or pollen grain is actually a male gametophyte. Microsporogenesis is a process in which microspores or pollen grains or male gametophyte is formed. So in this very process, we will see how these microspores, how pollen grains, or we can say how gametophyte is developed. So for this microsporogenesis, in order to understand the complete process of microsporogenesis, how the formation of microspores or pollen grains take place, we must understand the structure of anther. This is stamen. The complete structure is here stamen. In this stamen, I will label its components. This is
Filament.
Filament, and this is anther. These are two lobes of anther. Two lobes of anther. So we can say anther is bilobed in case of angiospermic plants. When we go through the anatomy of the anther, anatomy of anther. At its bud stage, there are epidermal cells, there are hypodermal cells, in the hypodermal cells. So I will discuss here the outermost cell layer is called epidermis. This is the first cell layer in the anther at bud stage. This epidermis is followed by hypodermis. Now we are going to discuss the microsporogenesis, the formation of microspores, which are also called as pollen grains, also called as male gametophyte. How the development of male gametophyte occurs, takes place. We have discussed here the structure of stamen. There are two important components. One is the stack-like structure that's called filament. Another is the knob-like structure that's called anther. When we see the anatomy of this anther, the outer epidermal cell layer is followed by another cell layer that's called as hypodermis. In this hypodermis, few hypodermal cells undergoes differentiation. So we can say there occurs differentiation of hypodermal cells. The end. The end करो.